Good morning, everybody. It is Monday about 8 a.m. and I cannot tell if it is hailing or snowing. It's a bit small and aggressive to be snow, but it doesn't seem to be large enough to be hail. So anyway, I am heading back in, as we discussed last week, to redo my cloning, which did not work. And today we are going to start off by discussing some strategies to add some new controls in, so that if something does go wrong again, we'll have a bit more of an idea about why. I have decided it is small but aggressive hail. I think I might have to explain controls on the way home. See you then. Good afternoon, everybody. A breakthrough. So last week my molecular cloning failed and I didn't really know why, basically because I didn't have enough controls in the system. So the goal this week was to redo the molecular cloning. We realized this morning when we went to get the plasmid out to start all over again that we probably used the wrong plasmid last week. So it looks like we accidentally used a very similarly named plasmid, which is why that probably didn't work. But we're still going to go ahead with the controls that we'd planned this morning because it's best practice to do that anyway. So my ligation's running at the moment, and tomorrow I'll be going in for my transfections again. But it is really nice to have a reason that we think it didn't work. I've at least got some optimism that it's going to work this time because I fixed the mistake, as opposed to just starting again and hoping it goes right this time. So I'll be doing FACS analysis tomorrow afternoon, Fluorescence Activated Cell Sorting. And then on Wednesday we will be imaging my cells for fluorescence and then also sending them away for sequencing. I'm really excited to see what this week has to offer. I'm very excited to have a go on the FACS machine. FACS machine. And I guess I'll go over that a little bit more tomorrow. See you then. Good morning, everybody. It is Tuesday morning. I do not know what time. I definitely had a bit of a lie-in this morning, so I need to get moving. So today's big goal is the FACS analysis. So I haven't really done FACS before, so I'm very excited to get into it. And then of course also today I will be taking off my ligation that ran last night. So I actually ran 4 ligations last night to include those controls we were talking about yesterday. So I'm going to get those ligations, I'm going to transfect them into the chemically competent cells, and we'll see if it worked this time. See you then. Good afternoon, everybody. What a day. So FACS analysis of my gene-edited cells looks very promising. It does look like we've had our intended effect. And it was very interesting to see that we tried 5 different guide RNAs. So guide RNAs are how you target your gene editing to a specific location. And it looks like each one of those acted at a totally different efficiency. So I think we got somewhere between 20 and 60%. For no particular reason we could see, just they acted differently. So it's very interesting to see. But we did expect it to some extent, that's why we had 5 in the first place. So we'll be going ahead with the best ones with our gene editing process. It's a beautiful sunny day here in Scotland for the moment. It has also been snowing intermittently. I got my transformations done as well today, the reattempt of the molecular cloning from last week, and we'll find out tomorrow if they grow on the plates. So I have a lot more plates this time. I've got about 5 because I've got so many more controls to test if they worked at different stages. But basically today was jam-packed and fantastic, and it also seems to be working out, which is really nice. So I'm looking forward to tomorrow. Good morning, everybody. It is another beautiful sunshiny day, and I have received some very good news from my lab supervisor. The way our coronavirus restrictions work is that not all of our rooms are big enough for 2 people. So some of the machinery I'm not able to be trained on yet because 2 people can't be in the room at the same time. So unfortunately I won't be able to image my plates myself for a little while. But this morning my supervisor went in ahead of me and he has said that he is seeing fluorescence on the plates. So the plates I'm talking about with fluorescence are the ones that I'm trying to do genome surgery on. So I'm trying to edit to their genetic makeup. So being able to visualize this with your own eyes is really cool. As in, I don't just have like numbers and charts from FACS. I won't just have sequencing data. I can see that I have had an effect. And I think that is profound. Also, with my molecular cloning that we were repeating, we have had colonies come up on the plates, which is also amazing. So this is the repeat of the molecular cloning that did not work last week. We'll have to go back in and see what we did differently. But mostly, we really think the difference was selecting the right plasmid. So I'm going to extract some DNA, but I'm hoping to also get some footage of the plates and the fluorescence. If I do get it, I'll pop it in here. So I've also put in an order for a mini ImageJ tripod, so I'm hoping to be able to film while I'm actually doing things with my hands in the lab. I'd love to bring you guys inside tissue culture, show you what it looks like when I'm splitting cells or transfecting things or extracting DNA, which is what I'll be doing today. And honestly, DNA extraction, pretty routine, pretty standard, and I think it doesn't look anything like what people think it looks like. So I'd love to share that with you guys eventually. So if you're interested in that sort of content where I actually bring you inside the lab and show you what I'm talking about, let me know. Good afternoon, everybody. Settle the glasses. Don't want to get fogged. Today was pretty brief. Like I said, I'm not able to do the imaging and I don't quite have the photos for you yet. I will be getting them this afternoon, so I'll show you some pretty pictures in a bit. So all I really had to do today was a DNA extraction. So I've extracted the DNA from those cells that I edited. Tomorrow I'm going to do a PCR to amplify that DNA and then send that amplified DNA off for sequencing. So once I've got that sequencing data, I will have the FACS data showing my gene edits. I'll have the direct images showing my edits, and I will also have the sequence data showing my edits. So I also had colonies come up on the plate from my molecular cloning the other day, which is great. So I've taken 10 of those colonies from across some of the different plates and I put them in for an overnight culture. Oh, I'm getting foggy. And I've put them in for an overnight culture. The idea being that we will then sequence those ones as well and see which one of them is correct and go ahead with that one. So by go ahead, that's a bit complicated to explain. I might explain that one better when that actually comes up because I'm not going to be doing much with them for a little while. But stay tuned. Alrighty, other than that, I'll see you guys tomorrow. Good morning, everybody. Very simple day planned today. I am going in to do a mini prep. A mini prep is a kit that just gets plasmid DNA out of a bacterial culture, typically an overnight culture. So when I had my plates with bacteria growing on them yesterday, I had little single colonies. I grabbed a single colony and put it in a single 15 ml tube. Those 15 ml tubes then spent last night in 37 degrees, shaking a little bit, and that's enough time for the bacteria to grow up. So when I go in, I'm gonna pellet it down. So I'm gonna put those tubes in a centrifuge and I'll get a little pellet of my bacteria. And then I use this kit to get the plasmids out of them. Anyway, I'm here now, so I'll let you know how it goes. Hello, I'm back. So today what I was doing was taking my overnight cultures from yesterday. You take some of them and put them in a labeled glycerol stock, which allows you to put them in the freezer without exploding. And then you take the rest and you extract the plasmids from it. To extract plasmids from bacterial culture, there is a kit called a mini prep. So basically you're just following the instructions. I did get some footage of it, but I want to make it into a YouTube short so it's in vertical mode. But go check out my YouTube shorts if you're interested in that. I don't know, that's a very new style for me, so we'll see how I go at it. I was a little worried about my glycerol stocks. So I pre-aliquoted the glycerol out yesterday, and it seems like maybe it dried out a bit or something. It was very hard for me to resuspend the overnight culture mix with the glycerol. So I don't know, it seemed fine in the end, but I didn't want to pipette them too viciously either because they're cells. I don't know. So the idea is I send these plasmids that I've now extracted away for sequencing to make sure that they have the insert that I put in them, and then I can go back to the glycerol stocks and grow up whichever one turned out to be correct. So this is just a fail-safe along the way to make sure that my plasmid is in the bacteria that I go forward with. So today was pretty simple, even if it wasn't particularly quick. So I'll see you guys tomorrow. Good afternoon, everybody. Today I did not have to go into the lab at all, so I did not have a journey into the lab to film. But I thought I'd bring you guys along for my afternoon walk because it's much more interesting to listen to me talk with a moving background than if I'm just sitting in my office. Or at least I think that anyway. So this week I did get my real data, my FACS data, which is really encouraging. It's not perfect, but that's fine. And also my fluorescent images. I don't have the sequencing done yet. We're just waiting on some primers that we can amplify my DNA with before we send it off to sequencing. But I probably will duck home and show you those fluorescent images. I can't explain too much about why I've done what I've done, but I think they look really good, so I want to share them. So all I had scheduled today was a lab meeting in the morning. They tend to go for about an hour and a half. I think it was only an hour today though. we all just keep each other updated on what we're up to and what we've been doing. And it's a really good environment to ask for help from people who are doing similar things, a bit different but similar. So this morning at lab meeting I got to present my FACS data and my imaging data, and I was really happy with that. So all in all, I am really happy with how this week has gone. So the only scheduled event I had today was a lab meeting this morning, but I'm spending the rest of the day refining a macro I made for ImageJ. A macro is kind of like really basic code, and ImageJ is software that researchers often use for image processing. But I'll show you a little bit about that when I get home. Okay, so I'm just jumping in here to show you those fluorescence images that I've been going on and on about all week, but also show you a bit about making macros. So right now I'm pressing record. Basically, the computer is going to record everything I do in these next few steps as code. And now I'm just splitting the original fluorescence image into the two different color channels, so blue and yellow. Now I'm maximizing the intensity on those two images. I've got to play around with the yellow. There's a lot of background fluorescence here. So now that I've maximized them, I'm going to save each individual image so that I can put them into a publication so that people can clearly see the blue channel and the yellow channel separately. And once I've saved these two individual images separately, I'm going to recombine them to make this image. So there you have it. That is my week in the life of a scientist this time. I hope you enjoyed those fluorescent images at the end. If you've enjoyed this content, please like and subscribe and leave a comment if there's anything you'd like to see in particular. Thanks guys, see you next week.